So as you know from many of the meetings, that many of the large-scale sequencing effort has identified a lot of driver mutations and the frequent mutations, and we also know from the cytogenetics that a lot of aberrations. But if you look at the heterogeneity of the treatment outcome and drug response, uh, there is no connection so far made to link those genomic alterations to individual drug responses. So we thought, so many of the effort has been driven in the precision medicine to try to understand phenotype from genotype. So our approach is the opposite. So we take the phenotype, okay, this patient is responding or not responding, then we get back to the molecular features and the genomics and transcriptomics and proteomics to understand why, and we also try to exploit that information to treat our patients in the clinic. So that was the main goal of our whole studies. Our approach is to tailor individualized treatment strategies for the multiple myeloma patients. So the way we do it, we screen each multiple myeloma samples that we derive from clinic with all the oncology compounds approved and investigational, and with the same samples we do NGS, the exome sequencing, we do RNA sequencing, we do have clinical information, the karyotype information, and all the clinical parameters and outcome, and we also do proteomics. So what we try to do with all the information from different level of the system's biology to integrate the information and try to understand the molecular biology of the disease. And one important thing that we do follow up our patients over time. So we do take, during the 4 years time, we take multiple sampling. So we do test the real drug sensitivity landscape each time the patient is getting new treatment, and also we do follow the progression of the disease during the whole time. So it's very dynamic. And if I sum up with the major finding is that we do see, as the genomic landscape, is very heterogeneous. So we do see a heterogeneity in the drug response too. So we saw that, but broadly the patient could be categorized based on the drug response in 4 different subgroups. So one subgroup was very interesting in terms of they are very sensitive to many of the signal transduction inhibitors. So we looked into the data of survival. They actually had the poor survival. And we looked into the genomics, and then we saw that they have the highest mutational load, and they have exclusively altered DNA repair genes and also many signaling pathway genes. So they have activated many escape routes. And these are the very progressive patients, and these are the patients that has been gone through already multiple lines of treatments, and the doctor doesn't have any treatment for the patients anymore. So what our approach is, and what we have done for these patients, is we took the drug response data and the genomics data, and we saw, okay, this group of drugs are very active in the patient. So we combine the signal transduction molecules, the approved one, for example, Rapalogs, we have combined with existing proteasome inhibitors, and we do see a dramatic response in those patients. So we do see some patients go partial response, and some patient, and we also do another thing, that that very limited amount of drug is approved for one disease, and whatever the stage of the disease, sometime, except for the karyotype, many of the patient get same line of treatment based on the diagnostic phase or relapse phase. But it is very important to understand that not all the patient would respond the same way to the same treatment. So you have to, and the timing is the most important, and the sequence. So what our stake is that we do capture the sensory landscape each time the patient relapses. So we know exactly what the patient is responsive to the treatment at the very moment, and we do modify the treatment strategies, fine-tune the treatment strategies based on the response, and we have very good success in translating the information to the clinic, and we're very hopeful about it. main implication of our study is to do this phenotypic screening and combine the molecular information to exactly diagnose the staging of the disease and the right treatment at the right time for each of the patients. Precise treatments and individualized treatments for each patient, which will ultimately improve the treatment outcome in the myeloma patients.